Hello and welcome to the webinar. Today, we will talk about a new way to simultaneously profile multiple classes of non-coding small RNAs at a new levels of sensitivity and accuracy. First, we will briefly review small RNA classes and their functional mechanisms, along with their superior biochemical properties for biomarker applications, and why do we study them. Then we will explain the challenges of current methods to profile small RNA expression, and then how do we best profile small RNAs using new small RNA profiling technologies. Briefly, RNAs smaller than 200 nucleotides long are small RNAs, which includes microRNAs, tRNA-derived small RNAs, or tsRNAs. tsRNAs include tRNA half molecules, or tRNAs, and further processed tRNA-related fragments, or TRF. Pre-miRNAs, tRNAs, and small nucleolar snoRNAs have sizes smaller than 200 nucleotides long and belong to small RNA. Small RNAs do not have sufficient open reading frames to encode proteins as mRNAs do. They are therefore non-coding RNAs. During biogenesis, microRNAs are first transcribed as a primary microRNA transcript from the host gene, processed by DROSA DGCR8 process enzyme into pre-miRNA helping precursor, exported from the nucleus into the cytoplasm, and processed by dicer into short microRNA duplex. The guide strand is the mature microRNA and is loaded into RNA-induced silencing complex, or RESC, containing ago protein to suppress the target mRNA. There are two major mechanisms of microRNA silencing, that is, translational repression on the translation initiation complex and mRNA destabilization and decay. Now let's talk about tRNA. Mature tRNA sequence is folded into secondary structure resembling the characteristic shape of clover leaf. It has, from the 5' end, the D loop, anticodon loop, anticodon triplex, T loop, and acceptor arm for amino acid attachment. tRNAs are the most heavily modified of any RNAs, with close to 100 chemical modifications. These modifications are an integral component shaping up and tuning tRNA structure and functions. tRNAs are tightly compacted into L-shaped 3D structure. The canonical functions of tRNA is for protein translation. tRNAs are encoded in nuclear and mitochondrial genomes. tRNAs are the most abundant small non-coding RNA. About 4 to 10% of total cellular RNAs is tRNA. tRNAs are the fundamental component in protein translation. They are accurately charged with corresponding amino acids by tRNA synthesis. Through the anticodon, tRNA decodes the genetic codon to translate mRNA sequence into protein sequence. Among the several codons for an amino acid, codon preference and usage reflect protein translational efficiency and accuracy. That is, tRNA isoacceptors have different anticodon and body sequences but carrying the same amino acid, and tRNA isodecoders have the same anticodon but different body sequences. In addition to the classical canonical protein synthesis functions, tRNAs also play regulatory roles. In the figure on the left, upper regulations of specific glutamic acid UUC and arginine CCG isoacceptors increases the pro-metastatic related mRNAs and leads to enhanced metastasis of breast cancer cells. In the figure on the right, an arginine UCO isodecoder out of 5 other isodecoders is expressed exclusively in the central nervous system. In the neurons deficient in this CNS-specific isodecoder, ribosomes will store at AGA codons. This storing can be rescued by GTP binding protein 2 or GTP-BB2. In the absence of both arginine UCO tRNA and GTP-BB2, severe ribosome stalling occurs, which leads to the stress response driven by activating transcription factor 4 or ATF4, neuronal cell death, and neurodegeneration. Many diseases, such as metabolic type 2 diabetes, cancers, neurological diseases, and viral infections by influenza, vaccinia, West Nile, encephalitis, HIV are correlated with major changes in tRNA repertoires. The profiles of tRNA isoacceptors and isodecoders repertoires are known to be involved in biological processes such as cell proliferation, differentiation, apoptosis, metabolism, and stress response. tRNA repertoires have dramatic causal or associative relations with human diseases such as cancer, diabetes, cardiomyopathy, viral infections, and neurodegenerative diseases. Now we will talk a little more about a new small RNA class, that is tRNA-derived small RNAs, or tsRNA. tRNA home molecules can be cleaved in the anti-codon loop region by angiogenin, which is open-up regulated upon cell stress conditions, hence the name of tRNA for stress-induced tRNAs. tRNAs are about 1/2 the whole tRNA size. They are also called tRNA halves, the 5' half and the 3' half. Angiogenin and dicer can further cleave the tRNAs into shorter pieces of tRNA-derived fragments, or TRF, producing TRF5, iTRF, TRF3, and TRF1 based on the fragment locations in the full tRNA. These specific cleavage products, or tsRNAs, are not random degradation products. They are a new class of small RNAs that have completely lost tRNA functions in protein synthesis and protein translation, but assumed new functions in biochemical regulation. tsRNA regulatory functions are often by mechanisms of targeting mRNAs. In the transcript engagement model, tsRNAs can guide the AGO complex to engage target mRNA the same way as microRNA. In the transcript displacement model, a tsRNA can bind and displace the protein factor, for example, YBX1 protein, from the mRNA to turn off the RNA binding protein effects on the target mRNA. There are many transcript engagement examples of TRFs acting as microRNAs to silence target mRNAs. Like microRNAs, they have similar dicer-dependent biogenesis, AGO complex loading, and mRNA silencing. Some microRNAs cataloged in mir base are actually mapped directly to TRFs. TRFs are so-called microRNAs in disguise. The aberrant expressions of tsRNAs can be just like microRNAs in diseases. For example, chronic feeding of alcohol increases complex C3 levels and induces oxidative stress and angiogenic. The increased glycine TRF generated by the angiogenic silences sirtuin mRNA, or SIRT1. SIRT1 downregulation causes liver steatosis and liver injury. Here is another example of tsRNA acting as microRNA. Valine TRF5 acts as a microRNAs to target freeze-3 mRNA, FZD3, causing downregulations of freeze-3 receptor for Wnt signaling transduction pathway, and eventually the suppression of breast cancer progression. tsRNAs can bind and engage mRNAs to open up mRNA fold structures to facilitate mRNA translation. Here, leucine CAG 3' tsRNA targets two sites in ribosomal protein mRNAs, RPS28 and RPS15, to disrupt the mRNA structures and to enhance their translation. Inhibition of tsRNA reduces ribosome biogenesis and induces apoptosis of dividing cells in vitro and in human hepatocellular carcinoma. The target sites are conserved across species, suggesting mRNA unfolding by tsRNA is a common conserved mechanism. In the transcript displacement model, the terminal oligo-G motif, or TOG tag of tsRNAs can form intermolecular G quadruplex, or R4G, which interacts with and displaces the translation initiation complex of the mRNA from activating translation. Shutting down global protein translation initiation is a major tsRNA feature during cell stress response. The stored translation complexes are sequestered in stress granules in the stressed cells. In another example of transcript displacement model, TIFs derived from glutamic acid, asparagine, glycine, and tyrosine tRNAs are induced by hypoxia and can bind and displace YBX1 protein from the 3' UTR of multiple oncogenic transcripts in breast cancer cells. Without YBX1, the oncogenic mRNAs HMGA1, CD151, CD97, and TIM3 are destabilized and quickly degraded. The net effect is TRFs being tumor suppressors. Here is yet another example of transcript displacement. A set of tsRNAs are induced by retinoic acid and displace IGF-2BP1, a well-known mRNA stabilizer, from c-MYC mRNA. With the downregulation of c-MYC, the stem cells undergo cell differentiation. tsRNAs acting on mRNAs either by engagement or displacement to regulate protein translation have earned their name as the Swiss Army knife of protein translation regulation. In addition to tsRNA's roles in mRNA and protein translation regulation, tsRNAs can bind target proteins for other novel molecular functions. During apoptosis, mitochondria release cytochrome C to form apoptosomes. A 5' tRNA can sequester cytochrome C away to inhibit apoptosis. Here is a summary of tsRNA functions. A. acting as microRNA. B. Translation inhibition. C. Guide RNA cleavage by AGO-independent mechanism. D. Displacing proteins on mRNA. E. Pause ribosome during translation. F. Translation from internal ribosome entry sites, or IRAS. G. Apoptosis. H. p53 phosphorylation. I. Ribosomal RNA processing. J. Primer for reverse transcription during retroviral replication. K. Block retrotransposon reverse transcription. L. Transposon RNA degradation with AGO. At the cellular level, tsRNAs are a major player in cell stress response and in the formation of stress granule, as in M, and N in cell proliferation. O. Exported in extracellular vesicles, and P. Carrying epigenetic information in sperm tsRNAs to transmit diabetes from the father to the offsprings. With these many molecular functions, tsRNAs are involved in many diseases such as cancer, metabolic disorders, stress injuries, and neurodegenerative diseases. Now let's move on to small nuclear RNAs, or snoRNA. snoRNAs are localized in the nucleolus, about 60 to 30 nucleotides in size. There are two major families of snoRNAs: CD-box snoRNA family guides 2'O methylations of rRNA, whereas HACA box snoRNAs guide pseudouridine modification. Minor members include carbohydrate-specific RNAs, or scarRNA, and some orphan snoRNAs. Complex with proteins to form small nuclear ribonuclear protein to form snoRNP complexes. snoRNAs guide ribosomal RNA modifications, whereas scarRNAs guide small nuclear RNA, or snRNA modifications. In addition to canonical roles in pre-ribosomal RNA and small nuclear RNA modification and processing, snoRNAs are regulatory functions by forming snoRNA-derived RNAs, or sdRNAs, to act as microRNAs in mRNA silencing, alternative splicing, stress response, regulations of cholesterol homeostasis, memory and learning in nervous system, and diseases such as cancer, Prader-Willi syndrome, autism, and viral infection. Recently, snoRNAs have been shown to guide epitranscriptomic modifications of mRNA. In the figure on the left, snoRNAs U32A and U51 guide 2'O methylase fibrillary FBL to modify internal 2'O methylations in the mRNA. The modification stabilizes the mRNA but block the mRNA translation. In the figure on the right, without snoRNAs and fibrillary, the mRNA is unmodified and less stable. However, paradoxically, the translation efficiency is greatly increased to make higher levels of the protein. One of the motivations to study small RNAs is using small RNAs as biomarker. MicroRNAs, colored blue, are abundantly present in biofluid, plasma, bronchoalveolar lavage, BAL, and saliva. tsRNAs, colored yellow, are predominant in ovarian follicle fluid, serum, saliva, cerebrospinal fluid, CSF, and massively in amniotic fluids, seminal plasma, urine, and bile. MicroRNAs have been popular biomarkers already. tsRNAs have been more attractive properties as a new class of small RNA biomarkers. That is, tsRNAs are heavily modified to render them resistant to RNAs degradation and become more stable, highly enriched in exosomes and extracellular vesicles, even more so than microRNAs. tsRNAs are pathologically related to stressful disease conditions, for example, cancers. tsRNAs display high degrees of tissue and developmental specificities. The abundances of tsRNAs depends on disease conditions. The tsRNA profiles have the power to discriminate triple positive or triple negative breast cancer cells from the normal cells as biomarkers. The whole molecule tRNAs have been difficult to profile in the past due to the technical difficulties to overcome heavy tRNA modifications and tight tRNA structures by small RNA sequencing. With the advances on tRNA profiling techniques, tRNAs are now emerging as a new class of biomarker. tRNAs have many desirable characteristics that make them a good biomarker. That is, there are abundant small molecules, small RNAs, and particularly enriched in biofluids, even more so than microRNAs in certain biofluids, such as saliva, cerebrospinal fluid (CSF), amniotic fluid, seminal plasma, urine, and bile. In exosomes, tRNAs are enriched 70 times, way more than microRNAs. The high tRNA abundance means higher biomarker sensitivity. At a cellular level, different tRNA repertoires have very distinct signatures for differentiating or proliferating cells. The differences in tRNA pools are primarily controlled by epigenetic histone marks on the tRNA genes. Likewise, snoRNAs have been studied as biomarkers in cancers such as breast, prostate, meningioma, and non-small cell lung cancer, NSCLC, either with tumor suppressor, TS, or oncogenic roles, OG. Now we have come to appreciate the biological and clinical significance of studying small RNAs. Profiling small RNA expression in the cells is often the first step. Due to the special biochemical properties, transcriptome-wide small RNA profiling has been challenging by small RNA sequencing. Small RNA sequencing involves many steps for CDN library construction, which accumulate and amplify biases and errors. These steps include small RNA pretreatments. Due to the small sizes, small RNAs require adaptive ligation at both 5' and 3' ends. During biogenesis, small RNAs may not always have the 5' monophosphate group and 3' hydroxyl group necessary for adaptive ligation. 5' end decapping, phosphorylation, 3' demethylation, dephosphorylation, and internal demethylations are required. During cDNA synthesis by reverse transcription, RNA structure interference, RNA modifications, particularly N1A, N3C, and N1G, and GC-rich contents in the RNA sequence can stop the reverse transcriptase and cause abortive cDNA copying. The cDNA library is PCR amplified, which distorts and skewers the representations of native RNA abundance levels by preferential cDNA sizes and GC contents. Another caveat of small RNA sequencing is the RNA abundance and differential expression analysis, commonly use TPM, or text per million rates. With TPM, if one microRNA level is changed, the TPM values for all other microRNAs in the total 1 million rates will also change proportionally, even if their absolute expression values are unchanged. In this simulated example, only does one start microRNA colored blue is doubled. All the TPMs calculated for other unchanged microRNAs will be reduced simultaneously in that total 1 million TPM. This is obviously misleading, and the calculated differential expression is erroneous. A much better way is to use Arraystar Small RNA microarray to do the small RNA profiling. In the workflow of the small RNA microarray, the RNAs are 3' dephosphorylated by T4 polynucleotide kinase (PNK) to remove the phosphate group P and cyclic phosphate group CP from the 3' ends to expose the hydroxyl group at the 3' end. The fresh 3' hydroxyl end is then ligated with silane-3 label by T4 RNA ligase. DNSO helps to denature RNA folds to reduce local RNA structural effects. The labeled RNAs are hybridized onto small RNA microarray containing entire transcriptomes of microRNAs, tsRNAs, pre-miRNAs, tsRNAs, and snoRNAs. Using direct enabling of RNAs for the small RNA microarray has many advantages. One enabling chemistry for all small RNA classes, compared with by sequencing that requires random primed reverse transcription for tRNA or snoRNA, yet adaptive ligation primed reverse transcription for microRNAs and tsRNA. There are no needs for small RNA class isolated by site selection, reverse transcription, PCR amplification that introduce biases and quantification errors. Simple DMSO denaturation is sufficient to minimize the effects of local RNA structural effects and distance differences on end labeling at the ends. Internal RNA modifications do not affect the enabling. Internal demethylation is no longer required. Minimal and efficient sample manipulations in the workflow. No sample amounts requirements as little as 100 nanograms of total RNA. Less vulnerable to chemically damaged or degraded RNA samples, suitable for archived or chemically treated samples, for example, FFP or biofluid RNAs. All this translates to faithful and accurate representations of native RNA levels. Several features have been incorporated in the array probe design. As mature microRNAs and tsRNAs have defined non-random 3' ends, the probe design includes a hairpin cap at the 5' end, which allows the perfectly aligned mature RNA ends to hybridize, while destabilizes and restricts the hybridizations for unintended precursors or isoforms, having longer extended ends. This design feature increases the end specificity to 1 to 2 bases. Furthermore, as the small RNAs are labeled with silane-3 label, the probe has a G at the fixed position to base pair with the C silane-3 that is always present at the 3' ends of the labeled RNA. This stable GC clamp significantly increases the hybridization signal. The probe target regions are optimized and normalized for uniform hybridization. For longer small RNAs such as pre-microRNAs, tRNAs, and snoRNAs, the probes are designed to target their unique sequence regions unhindered. The microarray contents include the collections of entire transcriptomes of cataloged microRNAs, tsRNAs, pre-miRNAs, tRNAs, and snoRNAs in the most current databases, such as MIR-base, tRNA-DB, MIMP-base, gtRNA-DB, Ensemble, as well as small RNA studies published in scientific literatures. The multi-transcriptomic small RNA profiling data are analyzed with the abundances and differential expression, richly annotated with small RNA biotype information, such as pre-microRNA genomic locations, microRNA target seed regions, biogenesis origins. tRNA isoacceptors and isodecoders, snoRNA CD box or HACA box types, statistical analyses, clustering heat maps, volcano plots are provided to screen and visualize and identify the most interesting differentially expressed small RNAs. In the research roadmap forward, the differentially expressed small RNAs identified on the small RNA arrays are confirmed by qPCR. The validated top differentially expressed small RNAs can be studied for their biological or pathophysiological functions by gain of function, for example, recombinant overexpression, synthetic mimics transfection, or by loss of function, LOF, for example, by antisense inhibitors in cell lines, animal models, or clinical samples. The molecular mechanisms can be studied for small RNA interaction with the target RNAs, for example, by bioinformatic targeting prediction, interfering RNAR silencing, AGO binding, or reporter assays for the regulatory effects of the targeted regions. The functions of the majority of the proteins are known and are cataloged. The proteins interacting with small RNAs are often indicative for the perturbed functions of the differentially expressed small RNAs. The small RNA protein partners can be studied by alternated RNA pull-down and are identified by tandem liquid chromatography and mass spectrometry, or LC-MS. The differentially expressed small RNAs are a good source of biomarkers, as we discussed earlier. The diagnostic biomarker performance can be evaluated by receiver operating characteristic curves, or ROC. The prognostic biomarkers can be studied by Kaplan-Meier survival or Cox regression analyses. To sum up, Arraystar Small RNA microarray is the new tool of choice, great for simultaneous multi-transcriptomic profiling of the small RNA biotypes, for example, microRNA, pre-microRNA, tRNA, tsRNA, and snoRNA. Higher standard of sensitivity, specificity, and accuracy for quantitative small RNA profiling. Direct and simple procedures to avoid small RNA modification interference, RNA structural hindrance, reverse transcription blocks, PCR amplification distortion, and quantification inaccuracy now being troubling for small RNA sequencing. RNA sample amount requirements as low as 100 nanogram, opening up many research opportunities, and tolerant for RNA samples at lower qualities, for example, degraded RNAs, serum plasma, biofluid RNAs, and FFPE RNAs. I think I will stop here. Thank you for your interest. Please visit us at arraystar.com.